Hi, I'm Alan Green, and welcome to Crash Course Biology. Today we're going to be talking about nucleic acids and enzymes. Nucleic acids are one of the four building blocks of life, and it's made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Nucleic acids are made up of a sugar, a nitrogen base, and a phosphate group, and together they make DNA and RNA. Nitrogen bases in DNA are adenine, guanine, cytosine, and thymine. Adenine and thymine pair together, and then cytosine and guanine pair together. In RNA, though, thymine gets replaced by uracil.
So to go into more depth of a nucleic acid, we know that there are three molecule groups that help make DNA and RNA. DNA is a double-stranded helix with nitrogen bases like thymine, adenine, guanine, and cytosine. This forms a nucleotide, which is a monomer of DNA and RNA. DNA stores hereditary info, also known as genetics. RNA is close to similar in a sense, but instead of obtaining a double-strand helix, RNA has a single-strand helix due to its function of forming proteins, as well as RNA containing uracil instead of a thymine and a nitrogen base. RNA sends information from DNA to ribosomes.
You want to hang out sometime?
Sorry, we just don't pair well together.
Hey.
Oh, hey.
DNA stores genetic information, while RNA, it helps create amino acids, which create enzymes. Enzymes are a type of protein, which is a catalyst, which basically speeds up reactions by lowering activation energy. The shape of the enzyme determines its function. Let's go to the top level.
Chaperones are a type of protein that folds enzymes. The cap comes off, the enzyme goes in, the cap goes back on, and it gets shaken up. Once shaken, the cap comes off, and the enzyme comes out folded.
As I said earlier, the shape of the enzyme determines its function, and there are three things that can mess up that shape. Its pH, the temperature, and the concentration. Now, let's go to the secret compartment. Let's see what's in here. Oh, graphs. My favorite. As you can see, the graph of the temperature. Once the enzyme hits a certain optimal temperature, it will start to denature and fall. For the pHs, there's a certain optimal range that the enzyme can stay in. Once it falls out, then it will denature. The concentration one is a bit different though. It will hit a certain concentration, the optimal, but it won't denature at all, it will just stay there.
I love enzymes.
So, a substrate is the thing that gets bonded with the enzyme at an active site, which is the active site is the point where the enzyme and the substrate will meet, and that's where everything goes on.
Enzymes are protein macromolecules. An example of an enzyme protein function would be lactase. As said before, enzymes act as catalysts to chemical reactions by speeding up the rate of reaction by lowering the activation energy. Enzymes have four levels of protein structure, the first being the primary level, consisting of linking amino acids making the protein. In a sequence, each group, R group, aka side chains, has its own unique function. In the secondary level, the protein gains a 3D shape from the hydrogen bonds and amino acids interacting with one another, resulting in an alpha A helix and a beta B pleated sheet. The tertiary level bonds with the side chains, or the R group together, of the amino acids within the structure. And lastly, the quaternary level is the formation of two polypeptides bonding together. So the normal protein and denatured protein are different because the normal is more compact and tightly connected than denatured protein, which is loosely stranded.
Okay. Thank you all for watching. This has been Crash Course Biology. Join us next week when we talk about lipids. And remember to stay awesome. Biology. Join us next week when we talk about lipids.
You know I wasn't recording, right?
Yeah, no.
Oh. Action.
This is gonna take forever.
What?
Wait, what?
I have to act like I'm already asleep. Hi, my name is Alan Green, and welcome to. Oh, I have a set crowd.